All right, let's dive into a scientific technique that's so powerful, it shapes our world every single day. The crazy part? It all happens on a scale that's completely invisible. We're talking about a microscopic sorting machine.
I mean, really think about that for a second. How do you possibly separate one specific type of tiny particle from a big messy soup of millions of other particles? It sounds almost impossible, right?
Well, the answer is this really clever technique called ion exchange chromatography. And believe it or not, this process is happening all around us, all the time. It's in industrial labs, sure, but it's also probably in the water filter in your kitchen. It is absolutely essential to our modern world.
So to really get it, let's start with the basic problem that scientists and engineers are always up against. Let's talk about the molecular sorting problem. You see, in the real world, the thing you want, whether that's a protein for a new drug, an amino acid for research, or even a nasty contaminant you need to remove from water, it almost never shows up by itself. It's always stuck in a crowd, mixed in with tons of other molecules.
So how on earth do you pull just one specific molecule out of that huge crowd? Well, you build a sorter that's incredibly picky, and you use one of the most fundamental forces in the universe to do it, electrical charge.
And that brings us to the official name, ion exchange chromatography. Now, ionic interactions sound super technical, I know, but it all boils down to a really simple rule we learned as kids. Opposites attract. That's it. It's basically like using tiny, microscopic magnets to sort molecules based on whether they're positive or negative.
To make this even easier, let's just think of the whole system as a molecular sorting gate. Imagine this tiny little gate that's designed to only let certain things through while catching others.
In science, this gate is called the stationary phase, and you can think of it as being coated with a kind of molecular Velcro. As the whole mixture of molecules flows past, this Velcro grabs onto any particle that has the opposite charge and just lets everything else wash right on by.
Okay, so the gate is great at catching the molecules we want. That's awesome. But that's only half the job, right? How do you actually get them off the gate again so you can use them? Let's walk through how it works.
This is the key question, really. It's not enough to just trap our target molecules. We need a way to release them, nice and pure, so we can actually collect them. The whole process happens in 5 main stages. First, you prime the sorter. You basically load up the gate with some weak, easy-to-remove placeholder ions. Second, you add the mixture, the whole molecular soup. Third, and this is the magic, you capture the targets. The molecules you actually want have a much stronger attraction to the gate, so they just come in and boot the placeholders right out of their spots. After everything else is washed away, you move to the crucial step, release and collect. You flush the system with a special solution that acts like a key, unlocking your target molecules from the gate. And finally, you just reset the whole thing and it's ready for the next batch.
So when you boil it all down, the whole thing is really just a constant competition. It's a non-stop chemical swap meet.
Okay, don't let the equation freak you out. It's actually really simple. On the left, you've got the sorter, S, holding a weak placeholder ion, E. Then your target, A, comes along. Since A has a way stronger attraction, it just swaps places. So on the right, the sorter is now holding your target, and the placeholder gets kicked out. It's just a simple, reversible trade.
Now, this molecular sorting gate isn't a one-trick pony. You can actually build it in 2 different flavors, depending on what you're trying to catch.
It's pretty straightforward. You can design the gate itself, that stationary phase, to have either a positive charge or a negative charge. This lets you zero in on exactly the kind of molecule you're looking for.
And the logic here is super simple. It's all about that opposites attract rule again. A cation exchanger is designed to catch cations, which are positive ions. So, naturally, the sorter itself has to be negatively charged. On the flip side, an anion exchanger catches negative anions, which means the sorter has to be positively charged. Easy, right? Okay, so that's the how. But now let's talk about the why. Why does this invisible little sorting process matter so much in our everyday lives?
Well, because being able to isolate specific molecules with this much precision isn't just a neat party trick. It's an absolute cornerstone of things like biotechnology, medicine, and environmental science. It's a total game changer.
And the applications, they're just everywhere. We're talking about everything from purifying the water that comes out of your tap, to analyzing the fundamental building blocks of life like proteins and DNA, to purifying the vitamins that go into your food. The list just goes on and on.
And get this, When it comes to water purification, this charge-based sorting is widely considered the single most effective method we have. That's a huge deal. It's what helps pull nasty ionic contaminants out of our water to make it safe to drink.
So, from life-saving medicines to a simple, clean glass of water, this process of sorting the invisible is quietly working behind the scenes. It really makes you wonder, doesn't it? What other incredible, powerful ideas are shaping our world, hiding right there in plain sight?